Well, we're back.
Hello.
With the lovely Jacqui Hardt, part deux, if you would.
Yes. For those of you that don't speak French, we are part two.
No.
It's definitely two.
It's definitely two.
Coming from an English person, it's definitely two.
So we are back and continuing on this trail. I think we just get right back into it. We were starting to talk about the different types of biological laboratories and their prevalence, pretty much.
Yeah, so biological labs are everywhere. I mean, academic institutions have many, many research facilities. If they have biology, biochemistry, veterinary, if they run those programs as degrees, they have those laboratories. And then you have many, many pharmaceutical companies, diagnostic companies, biologics companies, anyone who's doing research and development for pharmaceuticals, diagnostics, they will have entire facilities that are filled with biological labs. Then you have clinical facilities, anything that does diagnostics for hospitals or, blood test analysis, people who do things like genetic screening, they all have biological labs too. If they're running things on biological specimens from people, they will have a biological lab.
Can you give an example of that? So if I get a blood drawn, they shove it in a mouse and see if the mouse gets diabetes. How do they use the animal to test my blood?
So the majority of clinical specimens that are taken are used for diagnostics for human illnesses, diseases, cholesterol, pregnancy, whatever it is that person's doing. So they go to a clinical diagnostics lab, and usually it's just a test done on that clinical specimen to get the answer that test is running. Now, there are some circumstances where there is clinical research that involves animal models where that specimen could be directly introduced to a animal species to see, disease states or it depends on what the science is studying. They could also be used and then purified, and things could be pulled out of them, and then that could be applied to an animal model. They could remove pathogens from a human and apply them to an animal state to see how that animal behaves or reacts, especially if they're doing drug development to help prevent that disease. They expose them to that pathogen and see how the model species reacts to that therapeutic. So usually there's steps in between, but all of these types of facilities have biological labs. They are very prevalent. Now, animal facilities aren't so prevalent.
No, but what kind of animals are there? There's you got mice, I would assume. I'm thinking of the movies. Rodents. And you got monkeys.
Okay, yeah. So definitely from a live mammal perspective, mice and rats are your predominant species. But there could be research being done on other animals. Monkeys, there is monkey research facilities. They mostly deal with macaque monkeys, and they carry their own set of inherent diseases as well that can be harmful to humans if they bite you. So herpes B virus is one of those that can come from a infected monkey source.
Well, that's got to be an awkward conversation with a partner. I have herpes. No, I got it from the monkey at work.
This is herpes B. It's a different type of herpes. But yes, there are very unique risks for just animal handlers from dealing with unusual species. I mean, you can get mice and rat bites all the time as an animal handler. The tricky part comes in if they're infected with something. and then they bite you, right? It's a route of exposure. So what you need to know is that these facilities could be containing different species. They might just be a rat and mouse facility. They might have canines. I've visited canine facilities before. They are usually beagles that are used because that's the standard dog model. And the one thing that strikes you when you walk into a canine facility is the noise level. Those dogs bark.
Yes.
I mean, I did noise studies in canine facilities for worker exposure limit breaching because the barking is so loud. And, so they have very unusual hazards. Then there's also agricultural biosafety levels, laboratories, and they house large livestock. So if they're doing pigs or cows or horses or other large livestock mammals, especially for agricultural pathogens, then you'll essentially have almost a small farm that it doubles as a containment facility. I mean, they're going to have large stalls. You can't put a pig in a cage. You have to have an actual enclosure, a stall. Right. And it's a very, very controlled environment, especially if you have pathogens involved where those livestock are infected.
And I would imagine
to contain it.
The larger the container, the harder the control.
Mm-hmm. Yeah. So you have to consider the effluent that comes off that out of that facility. What do you do with the bodily releases of that animal?
Yeah.
Especially if it's infectious. What do you do with the bedding?
So does is that stuff neutralized on site? So from a hazmat transportation point of view, are those neutralized on site, or are they transported under like a DOT biomedical waste to a facility where they're neutralized?
I'm going to give you my favorite answer. It depends.
It depends. Right? That's our favorite answer as well.
Most often when it's BSL-3 or above, you're talking high hazard pathogens. They build into the facility a way of neutralizing that hazard before it leaves.
Okay. Bob's running a canine lab as well. It's a side project for him.
That's great. Now a monkey's going to jump across the screen, right?
Well, I have the monkeys. Yeah.
Oh, okay.
The monkeys are at my place.
Okay.
Yeah.
I'm hoping I don't have the rats at mine.
No. We don't deal with rats. They're not as much fun to play with. The dogs and the monkeys, at least you can play with while we're doing the experiments. It's really a lot of fun.
Refocusing. I am going to go back to the facilities that have these live animals that are higher risk. They tend to have autoclaves and things that they can deal with built into the facility. They're passed through. So they load them up on the hot zone, kill everything in the chamber, and then they unload it in a warm zone or cold zone. And then many companies still take the precaution of then transporting them as potentially infectious.
Okay.
There's a lot of validation involved with that process to make sure that the nasty is dead. Because when you're dealing with these high hazard pathogens, that's one of the permitted requirements for operating these facilities. And they have to follow very strict federal and state regulations for waste management. So they need to be in compliance with those.
So a first responder responding to a transportation incident that could have waste coming from, your standard precautions as a first responder would be sufficient, because if it hasn't been neutralized, it's a BSL 1 or 2, which really is not that high of a hazard in transmission, and your 3 or your 4s have probably been neutralized on site, and the transportation placarding is just a precaution.
Right.
Yeah, I mean, ones you really don't need to worry too much about. They're not generally generating biohazard waste. Twos often are more concerned than threes, in my opinion, because threes, they have these very stringent, established waste treatment procedures that they follow. But twos, those are still pathogens that can cause human disease. And usually they are just contained and shipped as regulated medical waste. Now, you also have the carcasses of these animals. So sometimes they die. Sometimes they are euthanized for procedural reasons for the science. And what do you do with the carcass? Well, there's also very stringent regulatory requirements as to what they can do with them. Most of the time, they're frozen because you start getting decay, and that can lead to obnoxious odors. So usually they ship out ratsicles. That's what they call them. And they have authorized waste haulers come in and manage the carcass waste. And it may be biohazardous carcass waste, depending on whether that animal has a wild type disease where, it's inherent to the species.
Okay. Or
whether it has been purposely inoculated with an organism for purposes of research.
So if you have a carcass that comes from a 3 or a 4, are they taken care of on site as well? Or almost all labs will ship the carcasses to whatever regulated facility is required by the municipality?
3s and 4s generally take care of their own waste streams.
Okay.
That they work with their local authorities to determine their efficacious. 2s could go out as standard UN 3291 regulated medical waste.
Okay.
They also have some carcass waste streams for non-pathogenic carcasses, which are managed slightly differently because they don't have the same associated level of risk. They're not biohazard waste. They're not infectious, just like, getting rid of a piece of chicken in your house, right? It's,
right.
They're just, and, each state has different rules. And so you really need to be aware of what your local jurisdictions require if that is your business.
Interesting. So, I mean, like in a non-emergency stuff, there's a lot of, if you're called for, let's say, a medical or just, an automatic alarm, like a smoke detector goes off or something like that. You're talking about, you got small animals, you got big animals, you got insects that could bite you, you got noise, you got all types of jazz that could be going on in there, right?
Yeah.
Besides the biological hazards.
Yeah. So you've got the organism hazards themselves. So you talked about insect labs. Well, usually insect labs are different facilities than, say, a mammal laboratory. Their research is focused on insects. It could be flies, very lot of like fruit fly research, very minimal risk if there is no pathogens involved. A lot of just genetic genome studies are done on fruit flies. But then you can also get into research that involves mosquitoes, ticks, spiders, crustaceans, centipedes, millipedes, and these organisms may have hazards just because they're wild type species can bite you or sting you. When you add the factor of pathogens in, I remember a company asked me once to do a risk assessment on a new research project that they were bringing into their space. And it was on West Nile virus, which is not a particularly pleasant disease. And it's a BSL-2 type scenario. So they had the facility to be able to handle that pathogen. However, turns out the room they were going into was next door to a mosquito research lab.
Oh.
So West Nile on its own, okay. Mosquito on its own, okay. West Nile virus with mosquito next to each other, not a good idea, because you put the vector for that disease in the immediate vicinity of the disease. And we're like, this is a terrible idea.
You're not gonna
inoculate your entire lab. Wow. If a mosquito gets out, somehow is infected with West Nile from your research next door, you're going to have an outbreak of West Nile in your lab.
Yeah.
With your people.
Or COVID if you're studying bats, Well,
that's the same principle, right? You don't want to put the vector with the disease, because that's how it spreads. There are tick-borne diseases, and you don't want the ticks near the pathogens that they can transmit, the Lyme disease, that kind of stuff. So
that sounds like kind of good housekeeping kind of thing.
Yeah, but at the same time, like if you're studying Lyme's disease and you're studying like transmission, how would you study that other than using a tick?
Tick model. Exactly. Right. So this is where you do a risk assessment. Yeah, they're very beautiful ticks.
Unbelievable.
Especially the research kind. So you have set up a facility. You guys, pull it together, please.
Sorry.
I learned that. I didn't know that.
That's what they call the organism of study, the model or the experiment. It's a tick model or a spider model or a mosquito model, whatever they're doing. That's the model organism.
Not like a GQ model. Totally. Yeah.
Wrong model.
An Abercrombie and Finch mosquito.
It would be highly attractive mosquito, but so they would risk assess, and they would then create a special containment facility, because that increases the risk. So that would probably go up to sort of BSL-3 type containment, where they would have very stringent controls to make sure that those infected arthropods, insects, are contained, cannot escape, right? You don't want them getting out. Number one, they need them for their research. Number two, you don't want, a Lyme disease carrying tick hopping, you might not even detect it. So containment. I know you mentioned, Bobby, like, what do you expect when you walk in these smoke alarm going off? Well, almost always the specimens will be contained. They'll either be in cages or they'll be in enclosures or they'll be in containers. The only time when they may be loose would be if a scientist is actually working on them or with them. If it's a very large animal, they likely won't be in direct contact. Now, they'll be in some kind of special enclosure or harness or cattle funnel. I'm not very familiar with, cattle housing terms, but
But at least
they want to contain the organism so it doesn't go rogue and cause them harm. So chances are, if you are going into a facility, there is a very minimal risk of a live organism being released. They might be anesthetized or unconscious, and they're usually using with very small specimen numbers at a time. They want to do the experiment on one thing at a time. So maybe one is out, but not like the entire 400 rats running around. It's not like a petting zoo where they're all free range. They are very running controlled. All over you. Yeah, that's it.
I know in the next episode, we're going to be talking about the emergency state. And we're continuing on the non-emergency state, but there's other things in the lab, like, controlled substances. I remember you were saying something about. What about that stuff?
Okay. So to perform these procedures on these scientific specimens, they often can't be moving. they have to be still. Sometimes they are doing like conscious research, things like behavioral studies. They're running around mazes and things like that in water, swimming. So they would put them in a special room with specialized equipment to be able to do that study. But very often they have to immobilize or even euthanize these specimens for performing necropsy, surgical procedures. So you can do that in a number of ways. Most commonly, you're going to use anesthetics. They could be actual injectable narcotics. Ketamine is a very common injectable anesthetic that is used to anesthetize live organisms or surgical procedures. And so there will be controlled substances because ketamine is a controlled substance. So it's controlled by the DEA. You will have to have a controlled substance permit in place from the DEA. And there will be a highly restrictive program surrounding the storage, use, disposal of that controlled substance in your facility. There will be very stringent security requirements. The anesthesia will be presented in a safe or a lockbox. There will be very strict inventory records, and you even have to do reverse distribution. If you have waste narcotics, you have to ship them back by an authorized distributor. So you essentially take custody of that controlled substance from the second it's ordered. And you can only order it if you have a approved license. And then you have to use that in a prescribed, approved protocol. You can't just go rogue and start injecting anything you want with that. The protocol itself is approved. And then you have to make sure to the end of life that controlled substance is entirely managed so it cannot get in the wrong hands. So yes, there's controlled substances in these facilities, but again, more often than not, they're going to be entirely secure and away from. There is a second form of anesthesia that is gaseous anesthesia. So isoflurane is a very common gaseous anesthesia that they use in anesthetic gas systems. They have a little, isoflurane comes as a liquid, and they put the appropriate dose of liquid into a special machine. They add oxygen, and then they pump it through nose cones, or they put it in an anesthetic chamber with a lid where they put the organism in, and that puts the organism to sleep for the duration of the experiment. Now, isoflurane will also put you to sleep. It is an anesthetic gas, and it has an occupational exposure limit. So I have done many industrial hygiene studies where the workers are being exposed to isoflurane, and they're nodding off as they're doing their surgery because they're also being exposed to the anesthesia. So one risk I would say is if you're responding to a medical emergency where someone's passed out in a space, a really good question to ask is, were they working with anesthetic gases? Because chances are they've anesthetized themselves. Plus, you have that risk when you walk in. You would have to wear respiratory protection for anesthetic gases to help respond to that situation. And that's independent of if there's a pathogen in there or not, right? So again, very unique situations. Now for anesthesia, they generally use carbon dioxide. Sorry, euthanasia. They use carbon dioxide. So there's compressed gases. There's definitely oxygen because sometimes they want them to wake up again and carry on with the study. So oxygen, obviously present in a fire situation, is not a good thing. So you want to make sure that you're aware from your fire department records whether oxygen is present. But then there's also carbon dioxide. And if somebody has been performing euthanasia procedures and they have gone unconscious, well, have they displaced the oxygen in the room and asphyxiated? So again, another hazard going in for a first responders is finding out like what it's the same as when we talked in episode 1. What's gone on in there? What were they doing? Because if they were working with carbon dioxide or with anesthetic gases, that could be the cause of the medical emergency. And you would need to know that going in.
The gases that you use, like carbon dioxide, oxygen, things along those lines, are they cylinder compressed or are they cryogenically compressed? And then follow up real quick, because I know you're gonna go, and then are those cylinders in a common space outside of the lab? Or if there was mitigation procedures that had to be done to those cylinders, are they inside the actual laboratory where you're going
to love this answer.
It depends.
Yes. All of the above.
All of the above.
It could be a cylinder inside the room with a piece of tube that they just turn the valve on and off. It could be a cryogenic liquid. They use liquid nitrogen for freezing tissues. So it could be a cryogenic tank of liquid nitrogen in the room. It could be a piped gas system where they are housed through the wall and they pipe it in. It could be a house gas system. So if it's a very sophisticated lab, they could have a giant tank outside in the parking lot that then it's a house gas system and they have taps. So all of those scenarios could occur, but they are all permitted hazardous materials, and it's very likely that they will be declared on hazmat permits, and to the fire department will charge these companies for having those compression systems on site. Lab gas permits are approved by fire department as part of building code. And then certain systems even have like air pollution control districts. So they're highly, highly regulated systems. So the chances are in normal operation, they won't go wrong. But you have the human factor here. Right. You forget to turn a valve off. Or
a natural disaster.
Well, we'll get there.
Yeah.
That's when the chaos. Some of them have, these facilities know these hazards, so they have real-time monitors, they have oxygen sensors, and it gives the workers heads up something's going wrong there so they can evacuate. But some labs don't. So, it just depends. Right.
Well, then we will leave it there for the non-emergencies. And next week, we'll be talking about the calamity and chaos that is ensuing. And that's why we're responding. So we'll see you guys next week.